After learning about Mendel's laws, our biology student might have thought that inheritance is neat and predictable, with simple dominant and recessive traits determined by Punnett squares. But then she wondered, if that's true, why do some traits, like her red-green colorblindness, not seem to follow those ratios Mendel described? Why is red-green color vision deficiency so much more common in males? And how did I get it? The truth is, many traits show inheritance patterns that deviate from Mendel's simple model. Hi everyone, I'm Noel Poller, AP Biology teacher and author of the Lab Hamster Storylines. During Unit 5, we've been exploring the story of a high school biology student who learned in class that she was colorblind and the family drama that unfolded after she revealed this information to her parents. In this video, we are going to explore non-Mendelian inheritance. These are patterns of inheritance that don't follow Mendel's rules that we learned about in the last video. I'll link that up in the corner right here if you missed that one. Okay, so what is non-Mendelian inheritance? Here's a preview of what you're going to learn about in this video. Non-Mendelian inheritance includes genetic linkage. This is when two different genes are located on the same chromosome. Codominance, when both alleles of a genotype are expressed. Incomplete dominance, when neither allele of a genotype can mask the other. Sex-linked traits, when genes on the sex chromosomes determine genetic characteristics. Pleiotropy is when one gene can determine multiple traits or effects. And non-nuclear inheritance, which includes traits determined by genes located in the chromosomes of mitochondria and chloroplasts. Let's start with genetic linkage. Mendel's law of independent assortment applies when genes are located on different chromosomes. But what if two genes are located close together on the same chromosome? In that case, the genes tend to be inherited together. They are said to be genetically linked. The frequency with which linked genes are separated by crossing over can actually be used to calculate the distance between them. This process is called genetic mapping and helps scientists determine how genes are arranged along a chromosome. Next, let's look at codominance. In codominance, both alleles in a heterozygote are fully expressed. A classic example is human blood type. Someone who has both the IA and IB alleles has type AB blood, where both A and B surface proteins are present on their blood cells. What about incomplete dominance? In this type of inheritance, neither allele completely masks the other allele, resulting in a blended phenotype. For example, in snapdragons, if a plant that has red flowers is crossed with a plant that has white flowers, the offspring plants will be heterozygotes and have pink flowers. These examples show that Mendel's dominant versus recessive model doesn't always explain the full picture of inheritance. Our student's color vision deficiency is an example of a sex-linked trait. The genes for red and green photopigments are located on the X chromosome. Since males only have one X chromosome, inheriting a single recessive allele for the condition is enough to cause color blindness. This is how Paul Anderson, or any other male for that matter with red-green color vision deficiency, inherited that trait. Females have two X chromosomes, which means they need to inherit two copies of the recessive allele to express color vision deficiency. This means color blindness is much more common in males than it is in females. By studying pedigrees, we can see these patterns emerge across generations. Blue-yellow color vision deficiency, on the other hand, is inherited differently. Since that gene is located on chromosome 7, it's not sex-linked. That's why blue-yellow color vision deficiency shows up equally in males and females. Another example of non-Mendelian inheritance is pleiotropy. This is when one gene influences multiple traits. For example, the gene responsible for sickle cell trait affects not only the shape of red blood cells, but also resistance to malaria, oxygen transport efficiency, and even risks for organ damage. In this case, we see that one gene can have many effects. Finally, some traits don't follow Mendel's rules at all because they're not carried on nuclear chromosomes. These are cases of non-nuclear inheritance. So where's non-nuclear DNA? The mitochondria and the chloroplasts have their own DNA. And in most animals, mitochondria are passed down only from the egg, which means mitochondrial traits are maternally inherited. Now, there are some rare cases of people who inherited mitochondrial DNA from their father, but that's extremely uncommon. In plants, both mitochondria and chloroplasts are passed down through the ovule, so organelle traits are also inherited maternally in plants. So while Mendel's principles laid out the foundation for our understanding of inheritance, the reality of genetics is much more complex. Genetic linkage, codominance, incomplete dominance, sex-linked traits, pleiotropy, and non-nuclear inheritance all show that heredity doesn't always follow simple rules. Thanks for watching. I hope this helped you see how inheritance can go beyond Mendel's model and all the factors that go into making us each genetically unique. So maybe you're wondering about identical twins. Do they always have the same exact traits? Or can other factors lead to identical twins sometimes having differences in their traits?